The old story was simple. Our species, Homo sapiens, appeared in Africa. We were smart, we had better tools, we had complex language. Sometime around 60,000 years ago, we left Africa and spread across the world. Everywhere we went, we met other older types of humans, the burly Neanderthals in Europe, the stout Homo erectus in Asia, and because we were supposedly better, we outcompeted them. We drove them to extinction. The story was a clean sweep, a total replacement. Our family tree was a neat straight line leading directly to us. That story was powerful. It was easy to understand, and for a long time it was the best explanation we had based on the bones and stones we found. But it was wrong. Not just a little wrong. It was fundamentally completely wrong. The real story is so much messier, so much more complicated, and infinitely more interesting. The truth, which started to emerge just over a decade ago, is that we didn't just replace them. We met them, we lived alongside them, and we had children with them. The idea of interbreeding wasn't entirely new. Some scientists looking at the shapes of ancient skulls had long argued they saw features that suggested mixing. A chin on a Neanderthal-like fossil here, a brow ridge on an early modern human there. But it was hard to prove. It was dismissed as seeing patterns in the clouds. The real proof couldn't come from dusty bones. It had to come from something much smaller, something hidden inside them. It had to come from DNA. For decades, getting DNA from ancient bones was science fiction. DNA is a fragile molecule. It breaks down after an organism dies. Heat, water, and microbes shred it to pieces. You were more likely to get the DNA of a soil bacterium or the archaeologist who handled the bone than the ancient human it belonged to. But a team led by a Swedish scientist named Svante Pääbo dedicated their lives to solving this problem. They worked in ultra-clean labs, developed new methods to piece together tiny, shattered fragments of genetic code, and slowly, painstakingly, they started to read the genomes of the dead. In 2010, they succeeded. They published the first draft of a Neanderthal genome, sequenced from bones found in a Croatian cave. The next step was the obvious one. They compared it to the genomes of people alive today. If the replacement story was true, there should be no overlap. Neanderthals were a separate branch that died out. Our branch kept going, they shouldn't have mixed. But they did. The team found that every person today whose ancestors came from outside of sub-Saharan Africa carries Neanderthal DNA. It's not a lot, usually between 1% and 4% of their total genome, but it's there. It's an unmistakable echo of ancient encounters. When our ancestors left Africa, they didn't just walk past the Neanderthals, who had been living in Europe and Asia for hundreds of thousands of years. Met them, and in some of those encounters, they had children whose descendants are alive today. We are all of us, except for those with the very purest African ancestry, part Neanderthal. This one discovery shattered the old story forever. Our family tree was not a neat line. It was a web, a network, branches split apart, and then thousands of years later merged back together. But this was just the beginning. The Neanderthals were just the first confirmed relative we had mixed with. The picture was about to get much, much bigger. Around the same time, in a cave in the Altai Mountains of Siberia called Denisova, archaeologists found a tiny piece of a finger bone. It was from a young girl. It was too small to tell what kind of hominin she was. It could have been a Neanderthal, since Neanderthal remains were also found in the same cave. It could have been an early modern human. The only way to know was to test its DNA. When Pääbo's team sequenced the genome from that tiny bone, they got a shock. It wasn't human, and it wasn't Neanderthal. It was something else entirely. It was a new, previously unknown lineage of hominin. They called them the Denisovans, after the cave where the bone was found. This was a stunning discovery. For the first time, a new type of ancient human was discovered, not from a skull or a skeleton, but from the genetic code in a fragment of bone. And then came the second shock. The team did the same thing they did with the Neanderthal genome. They compared it to the DNA of modern humans, and they found it, Denisovan DNA. But the pattern was different. It wasn't found in a low level across all non-Africans. Instead, it was concentrated in certain populations. People in East Asia have a little bit. But the highest amounts, up to 5% or 6% of their DNA, are found in people from Melanesia, people from places like Papua New Guinea, Australia, and nearby islands. This painted a new, even more complex picture. Our ancestors moving out of Africa first met and mixed with Neanderthals, likely in the Middle East. Then, as some groups continued to move east across Asia, they met and mixed with at least one group of Denisovans. This wasn't just a single event. It was happening in different places, at different times, with different groups of ancient people. So now we have two other hominins on the list, Neanderthals and Denisovans. That's a long way from a dozen, but the science of ancient genetics was just getting started. As the techniques got better, scientists could look at the DNA with higher resolution. They could see more subtle patterns, and they started finding strange things. They looked more closely at the Denisovan genome itself, and they found that the Denisovans had also interbred with another, even more ancient group. There were stretches of DNA inside the Denisovan genome that didn't look Denisovan, Neanderthal, or modern human. They looked much, much older. The best guess is that the Denisovans, long before they met us, had mixed with a super archaic hominin, something like Homo erectus, which had been living in Asia for over 1,000,000 years. We haven't found a Homo erectus genome yet, so we can't be sure. We just see its genetic shadow, its ghost, lurking inside the Denisovan genome. And since we have Denisovan DNA, it means some of us carry a tiny, secondhand echo of a hominin that might have lived 1,000,000 years ago. So, there's a third, a ghost super archaic. Then scientists started finding evidence of more than one kind of Denisovan. The Denisovan DNA found in East Asia seems to be genetically different from the Denisovan DNA found in people from Papua New Guinea. This suggests that our ancestors didn't just meet one group of Denisovans. They likely met at least two different, distinct Denisovan populations as they spread across Asia. So we can perhaps split the Denisovans into two groups. Let's call them Denisovan 1 and Denisovan 2. Now, our count is up to 4 distinct groups. We have ancestry from Neanderthals, 2 types of Denisovans, and a trace of a super archaic via the Denisovans. The story gets even more complex when we turn our attention back to Africa. For a long time, the story of interbreeding was focused on what happened outside of Africa because the cold, dry conditions in places like Siberia are much better for preserving ancient DNA. The heat and humidity of Africa destroys it quickly, so we don't have ancient genomes from most of the hominins who lived in Africa, but we can look for their ghosts. Just as we can find super archaic DNA in Denisovans, scientists can use clever statistical methods to look for strange, ancient-looking DNA inside the genomes of modern Africans. And they have found it. Several studies have analyzed the DNA of people from across West, Central, and East Africa, and have found sections of their genome that don't look like they came from the ancestors of modern humans. They look like they came from a much older lineage that split off from our own 500,000 years ago, or more. We don't know who these people were. Africa was a huge continent filled with different types of hominins. There was Homo heidelbergensis, maybe Homo naledi, and likely many others we haven't even found fossils of yet. The genetic evidence suggests that modern humans in Africa didn't just replace these groups. They met them and mixed with them too, and it wasn't just one group. The patterns of this ghost archaic DNA are different in different parts of Africa, suggesting that there were multiple distinct archaic populations interbreeding with our ancestors who remained on the continent. Some studies suggest at least 2 or 3 different ghost populations in Africa left their mark on modern DNA. So let's add them to the list. Let's be conservative and say 2 distinct African archaic groups. Now we're at 6 Neanderthals, Denisovan 1, Denisovan 2, a super archaic from Asia, and 2 African archaics. We are halfway to a dozen, and we are still just talking about the ones for whom we have direct or strong statistical evidence in our DNA. What about the ones we met but whose DNA didn't happen to survive in modern people? Or the ones whose DNA we just haven't found yet? Let's go back to Asia. The fossil record there is getting stranger and stranger. On the Indonesian island of Flores, archaeologists found the remains of a tiny hominin, barely a meter tall, that lived until as recently as 50,000 years ago. They nicknamed it the Hobbit, or Homo floresiensis. These tiny people lived on that island for hundreds of thousands of years. Modern humans arrived in that region around the same time the hobbits disappeared. Did we meet them? It seems almost certain. We were on the same island at the same time. Did we interbreed? We don't know. We haven't been able to get any DNA from the hobbit fossils. But the possibility is very real. Then, on the island of Luzon in the Philippines, another discovery. Fossils from another small-bodied hominin named Homo luzonensis, also dated to around 50,000 years ago. Again, right when modern humans were expanding through the area. Another potential candidate for an encounter. Another group we might have met, the Neanderthals and Denisovans themselves, weren't single, uniform populations either. They were spread over vast areas and over huge spans of time. A Neanderthal from Spain 100,000 years ago was likely quite genetically different from one in Siberia 50,000 years ago. When we say we interbred with Neanderthals, we are probably talking about multiple encounters with different groups of them. The same is true for Denisovans. It's likely there were many different populations of them spread across Asia, from the cold mountains of Siberia to the tropical forests of Southeast Asia. We have only found genetic evidence for 2 of them so far, but there were probably more. So let's tally it up again, not as confirmed species, but as potentially distinct populations that we encountered. 1, the main Neanderthal lineage we see in Eurasian DNA. 2, a second, distinct Neanderthal lineage found in the Altai Mountains that contributed DNA to Denisovans. 3, the Siberian Denisovan lineage we see in East Asians. 4, the Southeast Asian Denisovan lineage we see in Melanesians. 5, the super archaic ghost population that interbred with Denisovans like Homo erectus. 6, an unknown archaic ghost population that interbred with the ancestors of West Africans. 7, a different unknown archaic ghost population that interbred with the ancestors of Central African hunter-gatherers. 8, potentially another distinct archaic African group. The evidence is complex and still emerging. That brings us to 8 distinct groups for which there is some form of genetic evidence, direct or indirect, of mixing. Then add in the strong possibilities from the fossil record where we just lack the DNA for now. 9, Homo floresiensis, the hobbit. We were there at the right time. 10, Homo luzonensis. Same story, different island. 11, the red deer. Cave people from China. Fossils from as recent as 14,000 years ago that look like a mix of archaic and modern features. We have no DNA from them yet. And what else? There are probably entire lineages of hominins we have never found a single fossil of. They existed. They walked the earth. We might have met them. Their entire history is lost to time, unless a tiny fragment of their DNA happens to be found in our own genomes by future scientists. So is the number a dozen? It's not a crazy number. It might even be an underestimate. The key is to stop thinking of these groups as entirely separate species, like lions and tigers. The fact that we could have children with them, and that those children could have their own children, means we were all part of the same messy, interbreeding biological web. They were all, in a real sense, different forms of human. The human family was much more diverse than it is today. For most of our history, we shared the planet with other kinds of people. Today, we are the only ones left. But we carry the ghosts of those other people inside every cell of our bodies. This raises a profound question. What did we get from them? Was this ancient DNA just random junk that has stuck around, or did it actually do something? The answer is that it has profoundly shaped who we are. These weren't just random hookups. They were events that gave our species crucial tools to survive in new environments. Think about it. Our ancestors left Africa. They were adapted to an African environment. They walk into Europe and Asia, and they meet Neanderthals and Denisovans who've been living there for 300,000 years. These archaic humans were already adapted to the local climate, the local foods, and most importantly, the local diseases. Interbreeding with them was a genetic shortcut. Instead of waiting thousands of years for our own mutations to slowly adapt us to these new challenges, we could pick up useful genes ready-made from the locals. And that is exactly what happened. Some of the most common and powerful Neanderthal and Denisovan genes we carry are related to our immune system. Genes that help our bodies recognize and fight off viruses and bacteria. Many of these genes, called HLA genes, show clear signs of having come from our archaic relatives. They gave the first modern humans arriving in Europe and Asia an immune system boost, pre-adapted to the pathogens of their new home. It was a critical survival advantage. Another famous example comes from the Denisovans. Scientists were puzzled by how people in Tibet can live so comfortably at altitudes above 4,000 meters, where the air is dangerously thin. They found the answer in a gene called EPAS1. This gene helps the body manage low oxygen conditions. And the version of the EPAS1 gene found in Tibetans is not a normal human version. It's a perfect match for the one found in the Denisovan genome from that Siberian cave. The ancestors of modern Tibetans got this crucial high-altitude adaptation by interbreeding with Denisovans. Other examples are everywhere. Genes from Neanderthals affect our skin and hair, possibly helping our ancestors adapt to different levels of sunlight and colder temperatures. Genes from Denisovans have been linked to the ability of Inuit in Greenland to process fats from a marine-heavy diet. The genetic gifts from these ancient encounters are woven into the fabric of our biology. But it's not all good news. Our world is very different from the world of the Stone Age. A gene that was helpful then might be harmful now. Some Neanderthal genes that helped our blood clot faster useful to seal a wound from a hunting accident are now linked to an increased risk of strokes and pulmonary embolisms in modern people. Other bits of Neanderthal DNA have been linked to a higher risk for depression, nicotine addiction, and certain allergies. Our ancient inheritance is a double-edged sword. It helped our ancestors survive, but it contributes to some of our modern-day health problems. During the COVID-19 pandemic, scientists even discovered that a major genetic risk factor for severe disease was a chunk of DNA inherited from Neanderthals. 50,000 years later, these ancient genes are still affecting our lives and deaths. So, what does this all mean for our understanding of ourselves? It means that the idea of human purity is a myth. There is no such thing as a pure Homo sapiens. We are all hybrids, all of us. We are a mosaic, a chimera. Our genome is a patchwork quilt, stitched together from the DNA of our direct ancestors and the DNA of these other long-lost relatives. It also changes how we see our place in the world. We are not the inevitable, triumphant peak of evolution. We are the last survivors of a once diverse family. We are the only branch left on a bush that used to be thick with them. And we survived, in part, because we were able to take in parts of them to make their strengths our own. Our success was not just about replacing others, it was about absorbing them. The story is not over. We are still finding new fossils. We are still developing new ways to analyze DNA. We will almost certainly find more ghost populations lurking in our genomes. We might one day get DNA from a hobbit or from Homo erectus. Each new discovery will add another layer of complexity, another twist in the tangled story of our origins. The simple linear march of progress is gone. In its place is a much more real, much more human story. A story of migration, of meeting strangers in new lands. A story of conflict and competition, yes, but also a story of connection, of mixing, of finding common ground across deep differences. We are who we are because of these ancient encounters. The Neanderthals are not truly extinct. The Denisovans are not truly gone. They live on in us. A small piece of them looks out through our eyes, thinks with our brains, and fights off disease with our immune systems. We are the last of the hominins, but we carry all the others within us. The dozen is not just a number. It is our heritage.